the biochemistry. Okay, so how we are going to plan means we'll be discussing all the important topics from in 2 sessions. So biochemistry we are going to do in 2 sessions. Okay, so here first basically carbohydrates we are going to start. So carbohydrate, what is the only one thing you should remember? Your formulas, that is your CN H2NO, that's all. Okay, yeah, H2O and N. So this is your formula for your carbohydrates. And what is the N here? So N can maximum be between your 3 to 9, that's all. So next, coming to your disaccharides means when it is made up of 2 units, that is your disaccharides. Oligo means that is your 3 to 10, and poly means more than 10. So this is what they have asked once. Oligo means 3 to 10, poly means more than 10. This and all you know what. And actually, this is 3 to 10 is an controversial zone. Actually, ideally speaking, some books tell 8. 8 is oligo, more than 8 it is poly, but we can stick with this itself. This 10 and more than 10. That's all. So, and also one more nomenclature point that you should be knowing is aldose will not be having a UL. So, whenever there is a UL, that is called as what? Ketose. That's all you should be knowing. And next, coming to the isomerism. So isomerisms, what are all the points you should be knowing is just only one point: epimer. So what are examples of epimer? Constant example they will be asking, that is your glucose and galactose, and glucose and your mannose. Okay, so mannose has a 2N in it, so it is your C2 epimer, but galactose, it is your C4 epimer. But your galactose and mannose, they are not epimers with each other, because these two compounds will be differing in with respect to two asymmetrical atoms, no. And also 2 OH group orientation will be changed. So because of that itself, this is not itself an epimer. Next, coming to anomerism, that depends upon your OH orientation above the plane or below the plane, and your capital D, capital L, that is your enantiomers. So we shouldn't confuse between this enantiomers and your small d and small l. That is very, very important. And epimer, as I have told, it is nothing but your OH distribution with respect to your one asymmetrical atom. So these examples are very, very important point. Your glucose galactose and glucose mannose, very, very important point. And most stable form of your glucose is what? That is your beta D glucopyranose form. Okay, so glucopyrano ring, this is the most stable form. This is very, very important. And then anomer, I told no. So whenever your OH is above means what? OH is below means what? Whenever your OH is, yeah, below is alpha, and your above means your beta. So this is something you should be knowing it. And also enantiomers, they are nothing but mirror images of each other. So here what you have to choose means you have to choose the penultimate carbon atom. If it is right means that is your D, if it is your left means that is your capital L. That's all. So this is your isomerism for your the monosaccharides. And next, coming to disaccharides. So for disaccharides, only one thing only you should be remembering, that is what? That is your bond number, that bond number alone you should be remembering. And to start with disaccharides, first is your sucrose. Sucrose is nothing but a combination of your glucose and your fructose. So this you know already. So but they will be asking what is the form. So when they are asking about form of glucose and form of your fructose means, this is what? Alpha D glucopyranose and beta D fructofuranose. Okay, so this is very, very important. In your sucrose, it is in this form. So this is very, very important, and you shouldn't be missing it up. And next, coming to your trehalose. So trehalose, see, trehalose is alpha 1 comma 1. Just remember as a number, that's all. Alpha 1 comma 1 glycosidic bond. Maltose, so maltose is glucose plus glucose, but it is alpha 1 comma 4. So malto, it is your 4. Isomaltose, iso si, so it is for your what? Alpha 1 comma 6. Lactose, wherever you are seeing lactose, it has to be 1 comma 4. Lactose, it is beta 1 comma 4 glycosidic. And for lactulose is also fructose, it will not be having this thing, it will be having fructose plus galactose. Whenever it is glucose plus galactose, that is lactose. Lactulose means fructose, because it's a keto sugar, no UL group. This is also same linkage only, that's all. So this is for your disaccharides. Next, coming to your oligo. So oligo, already we have seen, it is 3 to 10. Okay, nothing, it is just some function, I mean your structural component, that's all. structural component of your cell membrane. That's all you need to know, nothing more than that. So next, coming to your poly. So poly means more than 10 by definition, and it can be more than 8 also, but always try to stick with 10. And here it is of two groups. So in this group, homo and hetero. Okay, our hetero is very very important for our questions. That's all. So coming to our homo, nothing, it is your glycogen, starch, mostly your storage polysaccharides, that is your glycogen and starch, that is your alpha glucose is made up of what? Alpha glucose units. This is very very important. And your okay, chitin. So chitin, when they're asking what is the main structural unit of your chitin, means that is your beta n nag, that is your beta n acetyl glucosamine, the glucosamine. So this is for your, most of the things will be in your alpha glucose. Alpha glucose is present in human or animals, okay, but whenever it is beta glucose, that is present in plants. So plants, the storage form is what? Cellulose. Okay, so this is very very important point. Now other than this, no other important point in the homopolysaccharides. And next coming to hetero. Hetero means all your agarose, okay, and your blood group antigen, okay, and pectins, dietary fiber, especially your pectin, gums, everything and all will be coming from your heteropolysaccharides. And most important is our focus will be around your mucopolysaccharides. Okay, so this is very very important, and all the time we can expect a question. So how to usually approach this kind of question means, see, mucopolysaccharides. So from the name itself you can see it's a polysaccharide. So polysaccharide means some sugar. So what is the sugar? That is your amino sugar. Muco means some uronic acid has to be there. So this is by definition some amino sugar with a uronic acid, it has to be there. And different types of mucopolysaccharides are there. Each will be having, so each will be having different exceptions. See, by rule you have to remember only two lines here. Only one mucopolysaccharide that is according to this rule is what? Heparan sulfate. But heparan sulfate, though it is like a good student, it is has one more MCQ here. It is neither acetylated nor sulfated. Okay, so this is very very important point. And next, coming to your, okay, in only in one more thing, your uronic acid is there now. Your uronic acid will be absent. So your uronic acid is absent in where? That is your keratin sulfate, both in your 1 and your 2. So 1 is present in your cornea, this is a question, and 2 is present in your cartilage, this is also a question. Okay, so this is how we have to approach this topic. And yeah, so next, coming to your hyaluronic acid. So hyaluronic acid, yeah, and also this functional unit is also very very important. See how you should approach this next, functional means there is chondroitin sulfate, there is dermatan sulfate. So C and D. So C and D, you can see it is chondroitin sulfate and dermatan sulfate, it is made up of similar units only. For example, N-acetyl glucosamine is there, okay, and also your glucuronic acid is there. That's all. Okay, so this is for your chondroitin sulfate, dermatan sulfate, keratin sulfate we know, heparan sulfate also we know. Hyaluronic acid. Hyaluronic acid, actually I made a mistake here. Your heparan sulfate, I said no, that is your neither acetylated nor sulfated. This point, yeah, that is for your hyaluronic acid. Yeah, heparan sulfate is glucosamine. It's made up of what? It is made up of glucosamine, that is your amino sugar, plus glucuronic acid. Okay, so that's all you should be knowing. And heparin, so heparin, how you can remember means, yeah, iduronate, one who, this line will help you. Yeah, heparin, one who injects heparin subcutaneously is an idiot. So this EDO is for your iduronic acid, that's all. Okay, and heparin is present in your mast cells, and other and all it's present everywhere. That's all. Dermatan sulfate, most widely distributed glycosaminoglycans. That's all. So what are all the pointers you have to remember is, most abundant, it is your chondroitin sulfate, we know. Yeah, so chondroitin sulfate, cartilage, bones and all, it is present. Next, no sulfation, no acetylation, that is your hyaluronic acid. And there is no uronic acid, means that is keratin sulfate. Iduronic acid, idiot, that is your heparin. And your dermatan sulfate, that's all. Okay, so these are your one-liner points you should be knowing. And also one more point you should be knowing is, This hyaluronic acid is no charge, no, because neither acetylated nor sulfated. No charge is here. Because of this, it can easily migrate. So cell migration is mainly because of hyaluronic acid. Corneal transparency, keratin sulfate 1, and your basement membrane, heparin sulfate. That's all you need to know for your basically the structural components of your mucopolysaccharidosis. Okay. Next, coming to your Hurler, Hunter, and all. Majority of the things you will be knowing. So, according to NEET, what will be the question? According to INACT, what will be the question? NEET, just know this corneal clouding point. Whenever without corneal clouding, then it is a Hunter. Hunter means what? That is iduronate. Okay, iduronate sulfatase. That is the enzyme deficient here. But Hurler means what? That is your L-iduronidase. Okay, L-iduronidase. That is the question in your Hurler, and Hurler will be having corneal clouding and all your mental retardation features and all. Next, coming to your Iniset. Iniset, the question is, so your Hurler is what? Type 1, Hunter is type 2, 2 for 2, 3 for your Sanfilippo. Most common they are asking, that is your Sanfilippo. And here it is N-acetyl glucosamine plus heparin sulfate. May or may not be important. And Marquis is for your floor. So floor means what? That is your 4. So Marquis, marble floor. So like that you should be remembering. And it is made up of your galactosamine plus your beta D galactosidase. Okay, lacto it is coming now. So here also lacto relation, that is your 4. That's all you should be knowing. This is very important point. And also one more point is how will you differentiate between, see, 1 and 2 differentiation, you know, that is by corneal. 3 and 4 differentiation, that is by using MR, mental retardation. So mental retardation is present in your Sanfilippo, it is not present in your Marquis. Otherwise it is more or less same only. Next, coming to your type 5 and your type 6. So type 5 is, that is your what? Shees. She is also similar to your L-iduronidase, that is similar to your Hunter, but it is a mild variety. That's all you need to know here. And Montrix larvae, it is milder form of your Marquis. That's all. M&M, that's all you need to remember. Last, this is something recent, so they can ask this, that is your Sly 7th type, that is beta glucuronidase. There is no mental retardation. So why this? Your top 3 will be having MR, your below 4 will not be having MR. So with this also you can easily guess here. So with the MR means only 3 possibilities you can diagnose. Without MR, Marquis, you know, that's a milder form, and there is the type 4 it is, and your type 5 is a milder form of your Hunter. And then coming to your Mortex lami, that is also a milder form. There is no questions here. Type 7 is important because the enzyme is very peculiar here, that is beta glucuronidase. Hope you got it. Okay, next, coming to your, okay, so these are your main introductions of your carbohydrates. So this is done. So short we can expect 1 to 2 questions, especially from your isomerisms and your other forms you can expect. So next, coming to your glycolysis. So glycolysis and all, see, coming to your metabolism. So the metabolism aspect is just, what can I say, some always easy scoring section is our metabolism. So in metabolism is concerned, first glycolysis. Okay, so glycolysis, what is the point you should be remembering? That is your rate limiting enzyme, that is your fructokinase 1. Okay, and what is the substrate level phosphorylation here? How many ATPs from substrate level phosphorylation? That is 4 ATP. That also you know it. And also, what is the aerobic glycolysis product? These and all, it's very typical question. Totally, if they're asking total, means that is your 9 ATP. If they're asking net ATP, that is 9 minus 2, that is your 7 ATP. This is for your aerobic. And whenever it is your anaerobic, means what? It is total, means that is your 4, but net means that is your 2. What is the main aim of your anaerobic? Is regeneration of your NADH, because this will tell the viability of the glycolysis reaction. Okay, so this is very important. And 3 irreversible steps. So what is the trick to remember 3 irreversible steps? Means just remember this GKHK. Okay, PFK1, that is your rate limiting enzyme, pyruvate kinase. That's all. And this difference between your GK and your HK is super important. Always you can expect, and this year also we can expect this question. So especially in this GK HK, you are able to follow now. Okay, so here HK is present in all the cells. This is one point you should be remembering. GK is present is only beta cells. That's all. And also GK. So GK is the isozyme. So it is also a type of this thing, but this is your type 4. Okay, this is your type 4, but your other thing, this is your HK is your 1, 2, and your 3 isozymes of your HK. This is important. And also one more thing. See, HK is present in all the cells. When it is present in all the cells, it should be having what? High affinity. Affinity is inversely proportional to Km. When there is high affinity, there is low Km. Exactly other way around here. That's all. And also the GK value. If they are asking some GK equivalent value means that is your 10 millimoles. But here, because the Km is very very low, the value is 0.05 to 0.08. This is around 10 to 150 or 180. This is the reason why the transport maximum of your glucose is what? 180. That's all. Okay. And also one more relation. See, insulin. What is the regulation of insulin on your GK and your HK? This is Ames level question. This is okay. So this point is always important. Ames level question. Your insulin is there now. Your insulin increases your GK activity. GK is an enzyme in glycolysis, so it increases this thing. And insulin via what? Usually all the insulinated enzymes dephosphorylation only. Okay, but on the contrary, no effect. Insulin has no effect on your hexokinase. This is the point they will be asking. One more point is G6PO4, that is your glucose 6 phosphate. Glucose 6 phosphate has no role in your GK, but your glucose 6 phosphate inhibits your HK. So this is the 4 points you should be remembering. Insulin action, G6P, glucose 6 phosphate action. Insulin increases GK. Insulin does not have any action over HK. G6PO4, that is a glucose 6 phosphate, does not have any action over GK. It will inhibit your HK. That's all you need to know. Next, coming to your PFK. So PFK already we know, that is your rate limiting enzyme and all we know. And in only one point here, what is it? That is your fructose 2,6 bisphosphate and your AMP. And another one is our fructose 1,6 bisphosphate. That's all. So always remember, though the number is more, this is a low energy state. But though the number is less, it is a high energy state. That's all. So whenever there is a low energy state, that will activate your what is the action here? So whenever your low energy state, that will be your PFK1 will be activated. But whenever in your high energy state, your PFK, there is no need for your glycolysis to happen, no, so that will get inhibited here. So this is very very important. So that's all. Next, inhibitors. Inhibitors, all the application orientations of all the cycles are very very important. First starting with inhibitors. So fluoride, that sodium fluoride is present in your what? In your test tubes, that blood collection for your gray tube, gray color vacutainer, it is present because it is going to inhibit your enolase. And where are we going to use it? We are going to use it for your gray vacutainer is used in diabetic patients. Okay, yeah, diabetic patients. And also arsenate. Yeah, this is very very important point. Other year, every other year they will be asking arsenate versus arsenite. Both are inhibitors only, but arsenate, nate is for your glycolysis inhibitor. Okay, and they will also be asking, is arsenate a direct inhibitor? No, it is an indirect inhibitor. Why? Because the intermediate formed is there. No, that is your 1 arsino 3 phosphoglycerate. That is the compound that is intermediate formed, that is going to inhibit your glycolysis. Okay. So next, coming to your, ultimately the end product of your glycolysis is what? Pyruvate. So next it is going to pyruvate dehydrogenase complex. The pyruvate dehydrogenase complex, only the coenzymes are important. So in the coenzyme, only one point you should see, there is PDH complex, there is dihydrolipoyl transacetylase, there is dihydrolipoyl dehydrogenase. So these are the enzymes that is present. What is the coenzymes that is needed means thiamine pyrophosphate, that is your B1. There is lipoic acid, that is your sulfur containing fatty acid. And your CoA has your B5. B5 is the coenzyme there, and FAD2 and your NAD. So NAD means that is your vitamin B3 and FAD means that is a vitamin B2. In this, what is the question they will be asking means B6. B6 is never a part of this PDH complex. That's all you need to know. So ultimately now the compound is entering into what? Mitochondria. Why? because of final regeneration of your ATP, and this is through your what TCA cycle. And in your TCA cycle, see just two steps, irreversible steps. What is the true steps? Same rule, follow the same rule. That is your citrate synthetase, sorry, citrate synthase, okay, not synthetase. Citrate synthase and your alpha ketoglutarate dehydrogenase. This is what you should be knowing. These two are your irreversible steps. And what is the net yield of ATP means? That is your 10 ATP. So for every molecule of acetyl CoA that is entering, we will be getting this thing. And if they are asking some total means, so total is your 34 ATP. When they are asking net, the net is your 32 ATP minus 2. That's all you should be knowing here. Anaerobic means same, 4 and 2. That's all you should be knowing. Okay, so this is very, very important. So next, coming to this inhibitors of your TCA cycle. Yeah, your inhibitors of your glycolysis is different, inhibitors of your TCA cycle is different, and this is where the questions will be coming. See, fluoroacetate, that is inhibiting your aconitase. Okay, arsenite, so nite for your TCA, nate for your glycolysis, and this is going to inhibit your alpha ketoglutarate dehydrogenase. You got the point, right? Yeah. So next, coming to your malonate. So malonate is what? This is very, very important. Succinate dehydrogenase. This we will be again dealing in the enzymes chapter also. That competitive, non-competitive inhibition part, that thing. So this is very, very important. So this sums up your fructose metabolism, glucose metabolism. So next, coming to your fructose metabolism, galactose metabolism. Fructose galactose metabolism, you can easily guess from the question. See, whenever there is a fructokinase deficiency, whenever there is a galactokinase deficiency. So in fructokinase deficiency means that is your benign fructosuria. Galactokinase deficiency means that is your benign galactosemia. Then what is the actual thing? So hereditary fructose intolerance. Hereditary fructose intolerance means aldolase B. When aldolase B is deficient, hereditary fructose intolerance, and most typical feature that is differentiating between galactosemia and your fructose is your hyperuricemia. Okay, so hyperuricemia differentiates that, and gouty presentations will be there. But in your galactose, that is your galput. So galput, whenever your galiput galactose 1-phosphate uridyl transferase that is deficient, that is your classical galactosemia, and the patients will be having mental retardation, organomegaly. These kind of presentations will be predominant there. And also your cataract is more towards galactose, and mental retardation is also more towards galactose, but your gouty presentation is more towards your fructose. That's all you need to know for this thing. So next, coming to your gluconeo. So gluconeogenesis mainly in your liver. Okay, so here what is it needed? So first, we know the three irreversible steps of glycolysis, like that is your pyruvate kinase, that is your phosphofructokinase 1, and then your GK. So how are you going to bypass these steps? So these steps are bypassed by three enzymes. So what are the three enzymes? Phosphoenol pyruvate, carboxykinase, fructose 1,6-BP bisphosphatase, and G6 phosphatase. So in this question, they will not be asking about what are all the irreversible steps, how it is bypassed. That question is outdated. We will not be having that. But then what is the question we can expect here is, I'll explain. Okay, so next, coming to your Cori cycle. So Cori cycle, always for a Cori is a child that is exercising. Okay, so Cori is exercising. So this is between your glucose and lactate. This understanding equilibrium between your muscle and your liver, that is it. And next, coming to your okay, propionic acid. So why propionic acid is important means, so propionic acid from what is the main source? That is your valine and your isoleucine, glucogenic amino acids. So next, coming to your propionyl CoA carboxylase is deficient means that will lead to propionic acid aciduria. Whenever your mutase or isomerase deficiency means that is L, this is important. Mutase or isomerase is deficient means that is your L-methyl malonyl aciduria. Okay, these comes under your propionic acid metabolism, so this is important. So what is the other amino acids? Means alanine means transamination. Okay, why this is important? This transamination is called as what? Cahill cycle. So Cahill cycle occurs during starvation. Your Cori cycle occurs during exercise. Cahill cycle occurs during starvation. Remember a child climbing up a hill and it is starving. So while starving, amino acids come for rescue, and this is a balance between your glucose and your alanine. And this reaction is called as transamination. That's all you need to know here. So next, coming to ultimately we are falling into HMP pathway. So HMP pathway, what is the main aim? The main aim is not your ATP. Then what is the main aim? That is your NADPH. NADPH, okay, not NADH. NADPH regeneration and ribose 5 phosphate. And where it is present? It is present in your RBCs. Why it is present in RBCs? Because RBCs doesn't have mitochondria. Okay, so it need but it need to survive, so it is utilizing this NADPH, and also to build this intermediate, I mean your materials, nuclear materials, we need this thing, substrates, that is your ribose sugars. That's all, nucleated precursors. Okay, so and the one important enzyme is there, that is your G6PD, that is your that rate limiting enzyme of your HMP pathway, and the RBC is going to survive by these enzymes. So whenever we are giving any drugs like sulfa group drugs or your fava beans, the patient will be having hematuria because of this G6PD lysis. That's all. So next, coming to your glycogen metabolism. So glycogen metabolism, two things, that is your glycogenesis, and another one is our glycogenolysis. Okay, glycogenolysis. So here first glycogen synthesis we can see. So synthesis, there is a primer protein. That primer is what? Glycogenin. So but the glycogenin is not active. So when it will become a primer is an active state, means it has to be added by 7 units. Okay, 7 units of your UDP glucose has to be added to make primer active. That's all. So ultimately it is forming linear branching. Linear branching is by forming by alpha 1 comma 4 bond. Branching is by alpha 1 comma 6. This and all you will be knowing very much. And yeah, this is something very very important. See, the rate limiting enzyme is glycogen synthase. Yeah, we know. There is two forms of your glycogen synthase. See, glycogen synthesis has to happen in energy rich state. In an energy rich state, your glycogen synthase has to be energy rich, means this is active, this is inactive. So this is your energy rich because glycogen can be formed here, but here it is inactive here. That's all. So that's all you need to know. And what is it mediated? It is CAMP mediated. This is very very important. Okay, this point, CAMP especially. So next, upon stress, they are asking, means see what is the effect of your insulin on your CAMP. So insulin on your CAMP is nothing but inhibition. Why? Because CAMP has to be inhibited, otherwise this will be happening. So CAMP has to be inhibited to activate this thing, glycogen synthase. Then only we can synthesize glycogen. That's the understanding you should be knowing. And the other compounds you should be knowing similar to your insulin is prostaglandin E1 and your nicotinic acids. Okay, so this is very very important. Next, coming to your glycogenolysis. So glycogenolysis and all you know it, but similarly the enzyme for glycogenolysis is what? Glycogen phosphorylase. That is the rate limiting enzyme. So the rate limiting enzyme of your this thing has two forms, two functional forms, bifunctional. That is your trisaccharide transferase. That can form also, that can break also. Break is by debranching. So two functions are there for this enzyme, especially your glycogen phosphorylase. Okay, so what is the MCQs surrounding glycogen phosphorylase? Means we know it is a rate limiting enzyme, yes, and we know it is active in phosphorylated form, yes, we know, because it is exactly opposite to insulin. And it require B6, pyridoxal phosphate. This is the first place we require B6 in our glucose metabolism. And alpha 1 comma 4 glycosidic bond activity, breakage activity is there, and it releases what? Glucose 1 phosphate. That's all you should be knowing here. Okay, and your okay, so next, what is the difference between glucose 6 phosphatase in liver and skeletal muscle? So in your liver, your G6PO4 is glucose 6 phosphatase is present, but here it is not present. Okay, that's all you should be knowing. And what is the final end point? So final end point is your G6P, glucose 6 phosphate, and because of this, in your skeletal muscle, the final end product is glucose 6 phosphate that has to be shunted again into your liver, and then it is entered into your glucose synthesis. Okay, gluconeogenesis or glucose synthesis. But the end product of your skeletal muscle is glucose 6 phosphate, because glucose 6 phosphatase is not present in your muscle. That is the MCQ they will be asking. Okay, so next, coming to your regulation. Yeah, this is the point I was trying to tell. See, glycogen phosphorylase is there. glycogen phosphorylase is there. Already it is a bifunctional enzyme, we know it. So phosphorylase, here also we need what? We need that CAMP dependent protein kinase, that we need it. Okay, but your glycogen phosphorylase is in inactive here, it is inactive here. Okay, and also always your CAMP is inhibited by your insulin. That's all. So for example, glycogen phosphorylase, that is a lytic enzyme. Glycogen phosphorylase is a lytic enzyme. Insulin, with respect to food, the patient is having insulin rise. That insulin is going to inactivate your CAMP. When CAMP is inactivated, glycogen phosphorylase is inactive, because that is what we need. If glycogen phosphorylase is active, the patient will be already sugar is high. Here the patient will also be breaking down glycogen. So then it will be tedious process. So that's all. Every biochemical process is properly well regulated. That's all you should be knowing here. And also one more thing is subunits: alpha, beta, gamma and delta. So there is your gamma is there. No, this has a catalytic activity. Okay, other things are not important. 100% catalytic activity. This is one subunit of your phosphorylase that has a catalytic activity. So next, coming to your other most important aspect, that is your glycogen storage disorders. So in our glycogen storage disorders type 0, this question is not that much asked uh, recently, but yeah, not that much asked recently, but whenever there's a glycogen synthase itself is positive. So glycogen synthase, that is the most preliminary step, is positive, efficient. Type 0 glycogen storage disorder. Von Gierke means type 1, that is your G6 phosphatase. So G6 phosphatase, anyway the muscle glycogen will be normal, muscle glycogen metabolism will be normal, because uh, G6 phosphatase is present only in liver and it is absent in your muscle normally. Okay, so this also it was a question, and here also the patient will be having what? Gouty manifestations, and there are two forms, that is your 1A and your 1B. Okay, 1A and 1B is there, and 1B, how are you going to differentiate? Means clinical features will be same, but in your 1B type the enzyme is normal. If enzyme is normal, then what is the actual pathology here? The receptor problem. Okay, here there is a receptor problem. That's all you need to know for your 1A and 1B. Type 2 is for Pompe. So Pompe cardiomegaly in your adults, that is the most important presentation, and also the presentation also. And in childhood it can be cardiomegaly also, but mostly it will be loss of or low muscle tone. Okay, and this is one GSD disorder, that is also a lysosomal storage disorder, and that's all you need to be knowing. And next, coming to your type 3. So type 3 is your 3A and your 3B. Okay, so 3A is liver plus muscle form, 3B is only your liver form. That's all. That's your what? Cory's disease. And it is also called as what? Limit dextrinosis. Type 4. Type 4 is Anderson. The baby will be dying in a very, very young age. It is also called as what? Amylopectinosis, because it is deposition of your amylopectin, and the baby will be dying of liver cirrhosis. That's all. So next, coming to your type 5. Type 5 is your McArdle, and this last NEET also they ask this question. And exercise intolerance and muscle biopsy, it shows what? Excessive glycogen. And this lab value is very, very important. So normally for you and me, it is less than 1% glycogen. But in your muscle biopsy of your this thing, it is your more than 4% glycogen will be present. This point is very, very typical. Unless otherwise, you mostly they will be giving this thing. What can I say? Your muscle biopsy picture. If they are not giving muscle biopsy picture, then they will be giving this value. You have to look for this value and then come to a diagnosis. So type 6. So type 6 is nothing but your HERS. HER is for your hepatic. So hepatic phosphorylase problem. Type 7 is for your TORI. So TORI is for your PFK1 enzyme. That's all. Okay. So how are you going to diagnose this TORI? So TORI means two things only will be present. There will be hemolysis, and also there will be exercise intolerance. That's all. So next, this is what? Hepatic phosphorylase. Then type 8. Type 8 is phosphoryl kinase. Okay. If some enzyme is happening, some enzyme is coming and you don't know where it is present, always choose liver. That's all. Okay. Type 9 is also same type, but that is present in liver plus muscles. That's all. Type 9, this is important. Type 9. Type 9 is very very important USMLE question, because that CAMP we saw now, CAMP protein kinase, that itself is going for deficiency. So which means that everything is normal in this apparatus, but everything is normal in your apparatus, but your CAMP protein kinase is the actual problem here. That is your type 10. Okay, I'm mistake here, this is type 9. Okay, so this is very very important, and this in this question, in this 10 things, type 10 is important. Phosphoryl kinase, phosphoryl kinase type 8, that is only in liver, means that is type 8. Liver plus muscle means that is your type 9. And also tori means two features, that is your hemolysis and your exercise intolerance. Her means hepatic phosphorylase, and your mcardle means your that 4 percentage, that value is important. Anderson amylopectinosis is also called as amylopectinosis, that is important. And your type 3 is your that limit dextrinosis, that point is also very very important. And also 3a also will be similar to what that we have seen now, that your type 8 and type 9. In your type 3a, liver and muscle, and in your type 3b it will be only liver. It will be exactly other way around. You got it? In type 8 also only liver, when liver and muscle is involved, that is your type 9 now. So like this way you should remember it. Okay, so this is how we should approach this thing. And one more point you should be knowing, that's a completion sake, is that is your uronic acid. So remember one thing, uronic acid can get converted into ascorbic acid, but it is not in humans, that's all. Okay, what is the enzyme that needed for this conversion is L. We know it's a glucuronic acid, no? So it is gulonolactone oxidase enzyme. Gulonolactone oxidase enzyme. They will be asking this. Humans cannot synthesize ascorbic acid in the uh, body because humans lack what? L-gulonolactone oxidase enzyme deficiency. That's all you should be knowing here. And also in this pathway only we'll be receiving that L-zellulose. So zellulose, essential pentosourea. So in essential pentosourea, what is the enzyme deficient? That is your zellulose reductase, zellulose dihydrogenase. Yeah, xylitol dehydrogenase. Okay, and whenever it is your d-xylulose reductase means that is your xylitol will be accumulated here. So whenever your d-xy problem is there, your xylitol itself will accumulate. Whenever your L-xylulose is accumulating, your xylitol reductase is present, is deficient, or your xylitol dehydrogenase is deficient. This you should be knowing. Okay, so next, so this completely sums your glucose metabolism. Got it? So continuing this with lipid metabolism. So lipid metabolism, there will be 3 to 4 regions only, mostly the questions will be coming. First question is your apoproteins, apolipoproteins numbers. Second is your dyslipidemias and your basics of lipids. And your last section is your fatty acid oxidation. Short. In this 4 subtopics, definitely 2 questions will be coming. Any 2. So starting with our basics here. So basics are first of all, lipid is a heterogeneous compound, non-heterogeneous compound that is soluble in your non-polar solvent. Okay, and there is a classification to it, that is your simple lipid, compound lipid, and miscellaneous lipid, and no one is going to ask question on that. And your phospholipids, yeah, see, phospholipid is a type of your compound lipid, and this is very very important. There are two types, that is your glycerophospholipid and sphingophospholipid. So glycerophospholipid means this combination is important. Glycerol we know, that is the triacyl glycerol we know, plus a fatty acid is going to be there, plus a phosphate group, plus a base. So this will be equation for your glycerophospholipid. But instead of your glycerol, when you are going to have sphingosine, that is your sphingophospholipid. Majority is your glycerophospholipid, minor is your sphingophospholipid. And bases can be nitrogenous or non-nitrogenous. Okay. So next, glycerophospholipids containing nitrogen, not containing nitrogen. So in this, whenever the name comes with phosphatidyl, Okay, mostly it is your nitrogenous lipid, except phosphatidyl ionositol. Okay, except phosphatidyl ionositol, that is non nitrogen. That's all. And also your cardiolipin, diphosphatidyl myoinositol, everything comes under non-nitrogen only. All the other things comes under here, and this is what they asked in last NEET. Phosphatidylcholine is lecithin. Phosphatidyl serine is nitrogen containing. Phosphatidyl ethanolamine is cephalin. So cephalin, your lecithin, your phosphatidyl serine. Fine. Next, coming to glycolipids. So glycolipids, see, this is very important point. Glycolipids, this is glycerolipids, okay? And next we are going to speak about glycolipids. So glycolipid is also a type of your compound lipid. Whenever instead of your phosphate group, we are going to have a glucose residue here. So this is nothing, saccharide, polysaccharide residue here. So basically it is of two types, that is a cerebroside and your globoside. So when there is a ceramide monohexo, it is mono, but it is your oligosaccharide, that's all. So this is the difference between your cerebroside and your globoside. And next you can see this ganglioside is there now. There is a ganglioside. This is the question. There is a GM1, there is a GM2, there is a GM3. So how are you going to remember? We know it is, yeah, ceramide. We know it. Ganglioside, it is a ceramide. So ceramide is going to come. Plus glucose is going to come. Plus galactose is going to come. Plus your N-acetyl galactose plus glucose. So this is how GM3 will be there. When your last glucose is not present, that is your GM2. When your last N-acetyl group is not present, till your glucose and galactose it is present with a nana, that is your GM1. So GM3 is the simplest thing that you have to remember here. So the equation I am telling it again. There is a GM1, there is a GM2, there is a GM3. So GM1 it is the most complex, GM3 it is the simple. That is the first question you should be remembering. Second thing is there is a ceramide that we know, there is a glucose, there is a we know, galactose we know, N-acetyl glucose we know, and your galactose also it is present in your GM1. Whenever your last glucose is not present, that is your GM2. Whenever your N-acetyl galactosamine that is not present, but instead of that we are going to have a nana, means that is your GM3. This is very important point. So next coming to your, so this is the basics for your lipids, one question short. So next coming to your plasma electrophoresis. So in plasma electrophoresis, depending upon your HDL and your chylomicrons, intermediately we are going to have VLDL and your LDL. Okay, high density. So high density, chylomicron is low density. High density, small size, low density, more size. So your chylomicron can fastly move because of low density. Okay, so this is something you should be knowing. So this will be the sequence. So your HDL corresponds to alpha band, VLDL corresponds to pre-beta, okay, and your LDL corresponds to your beta, and your chylomicron corresponds to your gamma. So gamma is fast. So that's all you should be knowing here. Okay, so next coming to your apolipoprotein. So apoprotein, mainly what is the main function of your apoprotein is to aid in lecithin cholesterol acyl transfer. So in this thing we need the function of, see, the next LCAT is a very important section, because how it is getting activated, how it is getting inactivated. So activation of your LCAT, it is by ApoA1, okay, and your ApoA1 and your lipoprotein lipase. So lipoprotein lipase is activated by Apo, remember, yeah, A1, C1, C2. This is you should be remembering. So this is for your activating your LCAT. But for inhibition of your LCAT, you should remember C2A3. Okay, C3A2, sorry, C3A2 other way around. It is C3 and your A2. So this will inhibit your lipoprotein lipase. So in a way it is inhibiting your LCAT. So this is how we should be delineating. So next, so this equation is very important. I am telling it again. Lecithin cholesterol acyl transfer, how it is getting activated, how it is getting inactivated. So it is directly dependent upon the activity of your lipoprotein lipase. So indirectly how it is getting activated, how it is getting inactivated. The numbers you should be keeping in your mind is A1, C1, C2, C3, A2. That's all you have to remember for your LCAT. Next, coming to your receptors and your tagged apolipoproteins along with it. So always remember HDL. So HDL, it is your A1, and your 100 for LDL, it is your ApoB100. So this is very very specific to each other, and all the other compounds will be having ApoE, the remnant variety. So your LDL receptor related protein, your VLDL, IDL, everything is going to have ApoE along with it, tagged along with it. That's all you should be knowing here. And also, so next important point is chylomicron metabolism. The basics of metabolism is not that much important, but one thing you should be knowing is your lipoprotein lipase. Your lipoprotein lipase is an endothelial enzyme. Okay, so endothelial enzyme, that you should be knowing here. And VLDL metabolism, similarly, VLDL, only one word you should be remembering. See, chylomicron, it is formed by absorption of your lipids from your diet, from the intestine. And then VLDL, it is synthesized in the liver. Mostly it is synthesized in your liver. And some breakdown product is called as what? IDL. That is also mainly targeted towards liver. And completely the end product is by LDL. So that is why it is getting deposited. And what is the main metabolism of your HDL means? HDL is reverse cholesterol acyl transfer. Okay, so that is why it is called as what? Good cholesterol. So in this thing, there is discoidal HDL, immature. So it is converted into spheroidal. So spheroidal is your mature type. So it has to acquire cholesterol from all the peripheral tissues, and then it has to take back again to the liver here. So this is very very important point. And yeah, when your there is a many lacunar spaces and all will be present. So when it is acquiring cholesterol units from the periphery, the lacuna will be sealed off. Okay, so the lacuna will be sealed off. So this is very very important point. Okay, and then next, cholesterol CETP, cholesterol ester transfer protein, and it is a plasma protein that is used for exchange of cholesterol units from the uh, tissues to the HDL and HDL to the liver. So this exchange is mediated by a cholesterol ester transfer protein. So what is the main function of this? It aids in reactivation of LCAT, and remnant particles assist HDL particle for the process of reverse cholesterol transport. That's also, this is like a motherboard for your HDL, your cholesterol acyl transfer protein. So this is very very important. So in this thing in this thing HDL2 to HDL3 type, so that is happening in the periphery. Okay, but HDL3 is converted into HDL2, means that is happening in your circulation. So this is very very important point you should be knowing. So this is for your basics of your complete lipid metabolism. Next, coming to your dyslipidemia. So before dyslipidemia, you should know one word, and this is very very important in AIMS point of view. The equation is called as Fradwall's equation. Okay, so Fradwall's equation. So what it is means, it is just nothing but see, only one thing you should be knowing: total cholesterol is equal to LDL plus HDL S plus VLDL. Yes, VLDL. So VLDL is equal to triacyl glycerol by 5. And in some textbooks you can use 3 also, but whatever feels comfortable with the values given in the question, you can stick with this. That's all you should be knowing here. And next, coming to your dyslipidemia. So your dyslipidemia, we have two things, that is your hyper, and next one is our hypo. Okay, so hyper not that much important, but hypo is very very important. Hypo can be of two types, that is ApoA1 deficiency or A beta lipoproteinemia. In A beta lipoproteinemia, A1 will be normal, and in A1 deficiency there is no HDL formation, because A1 is directly linked with your HDL. That's all you should be knowing here. And also, this, in they will be asking, in they will be giving an electrophoretic pattern, and when your HDL is what? HDL is your uh, high density means that is your alpha band. No, when your alpha band is not visible, that is your A1 deficiency. So like that they will be asking the questions. So this is for your hypolipoproteinemia. So your ApoA1 or A beta lipoproteinemia are examples of hypo. So next, coming to your hyper. So in your hypoproteinemia, first thing, the name itself is just remember the names, our job is done. So familial tri, arrow mark tri, glyceridemia. Okay, so see, from the name itself you don't need to remember anything. TGL is increased. When your TGL will be increased, your chylomicron will be increased and your VLDL will be increased. That's all. So next, coming to your cholesterol, second type is your familial hypercholesterolemia. So in this thing, there is cholesterol itself is increased. This is your LDL. So LDL clearance doesn't take place. So because of this, your LDL this thing will be increased here. So two things can be your pathology. One thing is LDL apolipoprotein ApoB100 problem, or your LDL receptor problem. That's also majority it is your receptor problem in your type 2. So in type 2, when type 2a type is there, type 2b type is there. In type 2a, your LDL clearance is affected. In type 2b, there is increased VLDL with minor LDL receptor problem. That's all you should be knowing for your type 2. So next, coming to your type 3. So type 3 is remnant removal disease, or your broad beta. So you can remember from the this thing, beta band corresponds to what? Alpha. Beta corresponds to your LDL. So LDL, that is your dis beta lipoproteinemia. That's all. So it is dis beta lipoproteinemia or broad beta disease or your remnant disease. So in this thing, we have three things, that is your E1, E2 and your E3. So 3 is, in this thing, your E2 will be always the question. Why? Because E1 and E2 easily recognized, clinically very recognized easily. So definitely that is not that much important. But E2 is very bad because it is recognized by receptors properly. Okay, so this is very important point. And what is going to be the profile here? See, this is the most dangerous. So your TGL, triacylglycerols, will be elevated, your cholesterol will also be elevated. That's all. And in your electrophoresis also, your beta band will be so broad. So see, directly you can go for diagnosis of your dis beta or your dis beta lipoproteinemia. So just summing up, type 1, type 2, type 3. So type 3 is your broad beta, your E1, E2, E3, that are all coming to play. E2 is our villain here. Type 1 is our TAG, that is a minor variety. Type 2 is hypercholesterolemia. In that we had type 2a and your type 2b, basically concerned with your LDL metabolism. Type 1a, we know that is basically your chylomicron and your VLDL problem only, basically. So next, coming to your type 4. So in your type 4 variety, it is your VLDL, actual VLDL problem. That's all. So type 5 is your deficiency of your ApoC. Okay, this will be similar to your type 1. That's all you should be knowing here. So next, coming to your familial hyper alpha lipoproteinemia. So this is nothing but alpha. What is your alpha? That is your HDL. So whenever your hyper HDL is present, that is called as alpha lipoproteinemia, and that is good to the body. Okay, and LCAT deficiency, lecithin cholesterol acyl transfer. So in this thing, from the periphery, the HDL cannot take cholesterol, lipids into the HDL molecule. Because of this, the shape will be remaining in your discoidal HDL alone. So this is very important. Hepatic lipase deficiency means that is your HDL2 levels will be increased, and your lipoprotein A is high means, yeah, this is very important. So see, small word only difference. When there is alpha hyperlipoproteinemia, that is good, but whenever that is your A lipoproteinemia is increased, which means that lipoprotein A is increased, means that has very atherogenic property. So that will result in stroke. So this is very important. Okay, why? Because your ApoA is present. No, your ApoA, it is a plasma protein, and it can gets activated by tissue plasminogen activator, and it can ultimately lead to your activation of your coagulation system and block thrombosis, stroke. Okay, so this is very important point. So summing up the disease, it will be very simple. See, whenever your ApoA is given in the question, definitely some thrombosis manifestation you have to suspect. Whenever your alpha hyperlipoproteinemia is present, your HDL, that is good for our thing, and your lecithin cholesterol acyl transfer problem means your discordal HDL will be remaining. And whenever there is your HDL2 levels is more means your hepatic lipase activity is hampered there. And defective ApoC, very minor variety, it is similar to your type 1. And VLDL2, 4 letters are coming, so it is your type 4. and your E1, E2, E3, E2, E looks like 3, so that is your 3, E2 is your bad, E1, E2 good. This is called a broad beta disease or beta lipoproteinemia. Hypercholesterolemia, so cholesterol word is coming means that is your 2. Okay, so basically it is your LDL. TAG will be your VLDL and chylomicron. So from the name itself, we can easily tell whether this is type 1A or 1, 2, 3, 4 like that. So triacylglycerol in your triglyceridemia means type 1, cholesterol means type 2, broad beta or beta lipoproteinemia means that is type 3, VLDL means type 4, ApoC means type 5, and your HDL2 level is elevated means that is hepatic lipase problem. LCAT means discoidal HDL, and your this thing apolipoprotein A means that is your stroke. So from these keywords itself, we can easily diagnose the question. That's all we need to know here. Okay. So next coming to your fatty acid oxidation. See, fatty acid oxidation, short point. Either way, they'll be asking it. So there are three types. First, fatty acid oxidation. So where it is occurring in your cytosol. Okay, fine. First question over. So most common fatty acid that is getting utilized is our 16 carbon atom. That is your palmitic acid. Okay. So in your palmitic acid, basically, here what is the thing they are asking is how many acetyl CoA is formed. So seven cycles means 7 acetyl CoA. In your last step, you will be ending up with 1 plus 1, that is a 2 acetyl CoA. So totally 9 acetyl CoA will be present. So if they are asking how many cycles means, in case of 18 carbon atom, you have to divide it by 2. That is your 9 plus 9 cycles, plus in your last step, you will be having 1 plus 1. So totally you will be having 10 acetyl CoA and your 9 cycles. This is how we should be remembering it, and this is very important point. And what is the precursor molecule is not acyl CoA, it is acetyl CoA. Acyl CoA is building block. Acetyl CoA is the precursor molecule for your fatty acid synthesis. Okay. So next coming to your fatty acid synthesis complex is there. In fatty acid synthesis complex, how many enzymes is there? 7 enzymes. So in this 7 enzymes, 7 catalytic domain. Okay. So acyl acetyl transacetylase, malonyl transacylase, so two transacylases, ketoacyl synthase, ketoacyl reductase, so two ketoacyls, hydratase, reductase, thioesterase, not thiophorase or thiolase, it is thioesterase. So this is very important for your, because all of the following are components of your fatty acid complex, except like that they will be asking the question here. So that's it, and you don't need to know about the details of any other thing. And the last enzyme that is involved is thioesterase. This is a question, 2 or 3 times they have asked this thing. And okay, this fatty acid synthesis complex enzyme maximum, it can utilize how many carbon atom fatty acids mean, that is 16 carbon maximum, it can utilize that only. When it is getting more or less, other mechanisms will be coming into the play. So basically this elongation is happening in endoplasmic reticulum lumen, and denaturation is also happening in the same place, endoplasmic reticulum lumen. It is especially for your, when it is more than 16C or it is less than 16C, very short means you will be having this. So this is very important point. So next coming to the actual steps involved in your fatty acid oxidation is fatty acid oxidation, 3 types: alpha, beta and gamma. So in this thing, alpha and gamma, there is no ATP production. Beta, it is ATP production. So beta is our examiner's favorite and our importance also. So first, alpha oxidation. So alpha oxidation, what it is, there is no ATP that we know it, basically for your branched chain fatty acids. So your branched chain fatty acids, especially your phytanic acids, plant sources and all, will be using this. That's all. So what is the main function that is happening in your alpha oxidation is your methyl group is removed. That's all you should be knowing here. So whenever there is a defect in this alpha oxidation, that will result in what? Refsum's disease. So whenever your methyl group is not removed, that will go and deposit in your brain, and that will result in mental retardation, and in your muscle that will result in your myopathies. So this is very important. And also one more feature that we have is blindness. It is concerned with your this thing, and muscle, we will be having extra phalanges in your hands. So this is very important in your Refsum's disease. So next coming to your gamma. So gamma oxidation, so endoplasmic reticulum, it will be occurring here, and in your alpha oxidation, we saw it. So in endoplasmic reticulum, it is basically for your medium chain fatty acids. Okay, and where it is occurring? It is occurring in your same brain and your white muscle fibers. Okay, so white looks like omega, no? So that's how we have to remember here. So white and omega looks similarly. So that's how we have to remember. And also this is that medium chain. So there is a dicarboxylic acids and all coming under this thing only. Okay, so dicarboxylic acid. Okay. uh, okay, fine. Okay, ultimately it is getting converted into dicarboxylic acid and then it is getting entered into your beta oxidation. So whenever there is a dicarboxylic acid levels are increased in the urine, which means what, till this step this is normal, but your beta oxidation is the problem here. So you have to evaluate the patient for beta oxidation defects here. So next beat, yeah. Yes, and also in beta oxidation, where it is happening. See, your gamma oxidation happens in, yeah, gamma oxidation happens in your endoplasmic reticulum. Your beta oxidation happens in your mitochondrial matrix. And when it will occur, mitochondrial matrix, okay, lack of insulin, because lack of insulin means energy deficient state, no. So energy deficient state only this will be happening. Main purpose is to replenish ATP. ATP production is the main this thing here. So your fatty acid is present, so that has to enter into your mitochondria. That one shuttle is there, that is called as what, carnitine shuttle. Okay, so here carnitine shuttle, the structure, this structure is very very important. There is a mitochondria, there is outer mitochondrial membrane and inner mitochondrial membrane. What is present in these things? There is a CPT1. Okay, there is CPT1, there is a CAT1. Inside inner mitochondrial membrane, we have CPT2, there is a CAT2, and also there is a CAT. We shouldn't confuse between CAT with our CAT2, entirely different. So CPT means what? That is your carnitine palmitoyl transferase. CAT means carnitine acyl transferase. Whenever there is a number is there, that is an enzyme. Okay. Other than that, without a number means that is carnitine acyl carnitine translocase. Okay, without a number means that is a translocase, with the number means that is a transferase. Okay, we shouldn't mess it up, that thing. And also bigger number is present inside, inner mitochondria number is more. That's all you have to know here. And then ultimately, for example, in your 16 carbon palmitic acid is undergoing beta oxidation, means it is ultimately 7 cycles will be formed, and your 8 plus 1 9 acetyl CoA will be left. So total will be your 108 and your net yield will be 106. So your brain and RBCs has these features, this ketogenesis feature is absent here. That is why they can't utilize ketone bodies in their this thing. So ultimately when the patient is going on exploiting his beta oxidation properties, means especially during starvation and all, that will result in ketone body generation. That is why we are getting what, ketosis in diabetic ketosis and all, we are getting this because of excessive activation of this cycle. So on this note, we are entering into what, ketogenesis. So genesis occurs in your liver, but lysis occurs in your periphery. Okay, so this is very very important point here. And also one more point that is very difference between your liver and your periphery is there is an enzyme called as thiophorase. See, thioesterase means that is a FAS complex, but thiophorase means that is a ketogenic enzyme. So liver lack thiophorase, hence acetoacetate CoA conversion is not possible. Acetoacetate to your acetoacetyl CoA conversion is not positive because in liver this thiophorase enzyme is not present, but this is present in your periphery. That's all you need to know. So next coming to, so now we are done with our branched chain, that is through your alpha oxidation. Now we are done with our medium chain, that is to gamma, and it is giving rise to a dicarboxylic acid, that is giving rise to your beta. So in your beta oxidation also, ultimately our ATP is synthesized now. So what happens in very long chain fatty acids? So in very long chain fatty acid, the main organelle is our peroxisomes. Okay, so in your peroxisomes, whenever this is getting, that's more than 24 carbon. So this is the definition, this also they have asked. So whenever there is more than 24 carbon and accumulation of and any enzyme deficiency is there, accumulation of this happens in your brain, muscle, liver and kidney. Okay, so this is the only place where your liver and kidney is coming into play. All the other places brain and muscle only. So main reason is peroxisome problem, that is lack, absence of peroxisome, or maybe empty peroxisome. Though the number of peroxisome is normal, it is defective, that is called as empty peroxisome, or your lack of peroxisome. These could be two reasons for your Zellweger syndrome. So this is called as cerebrohepatorenal syndrome. Cerebrohepatorenal. Okay, that is the name for the Zellweger. Refsum's disease is alpha oxidation, that is phytanic acid oxidase problem here. So this is very very important point. Okay. Okay, fine. And then, yeah, one more point I wanted to tell is, see, your beta oxidation is mainly concerned with ATP production that I told, yes, but your very long chain fatty acids, initial step is there, no. That initial step can occur or can undergo beta oxidation. Okay, later it will move on to your peroxisomes for your further this thing. So this is very very important. So beta oxidation is concerned with ATP production and also for initial steps of your very long chain fatty acid metabolism. So that's all you need to know here. Okay. Next, coming to your cholesterol synthesis. So cholesterol synthesis and all, no, basically no major questions will be coming for cholesterol synthesis, but you should be knowing that it is a 27 carbon compound here, and since it is a synthesis, it is active in your dephosphorylated state, and insulin is going to favor this cholesterol formation. That is why you shouldn't take too much of carbohydrates in your diet. So ultimate byproducts of this is your bile acids. So in your bile acids is concerned, what is the rate limiting enzymes? That is your 7 alpha hydroxylase. That is the rate limiting enzyme of your bile acids. So this is very very important. And also one more point I would like to stress here is, there is two forms. Okay, there are two main bile acids, that is your cholic acid and your cholic acids. So why is this important? Both are your stored in your gallbladder, that is fine. And there is a glycocholic acid and a taurocholic acid because of your conjugation with your glycine or your taurin. So whenever glycine comes into play, that is called as glycocholic, taurocholic, glyco deoxycholic, taurodeoxycholic. So that is the this thing, and everything can be precipitated with as salts also with your sodium and your potassium. Okay, so what is the main function here means, see, ultimately this bile salts, they will be entering into the duodenum. Okay, and in your duodenum and then into your terminal ileum. So in your terminal ileum, if like proper fat is present in the diet, it is going to enter into your take that and enter into your enterohepatic circulation. If this is not there in your terminal ileum, because of the action of your the conjugation has already occurred here, no, by using this taurin and glycine. When this bacteria action comes into play, this conjugated thing is converted into, it's deconjugated. Okay, two things happen that can happen is deconjugation, and another one is 7 alpha dehydroxylation. So when this happens to the bile salts, it is excreted into your uh, feces. That's all you should be knowing. And also this cholic acid, that is some part of it is converted into your lithocholic acid. So lithocholic acid is also excreted in your feces. So what are all the fates, like your cholic acid can be converted into lithocholic, and that can be excreted in the feces. That is one thing. And also one more thing is your this thing, your because in your terminal ileum, because of the action of your bacteria, it can get a d it can get deconjugated, and your 7 alpha dehydroxylation can also be happen and can be excreted in your feces here. And one more thing is, in your duodenum and your terminal ileum, when appropriate fat is present in the diet, it can take this and enter into the enterohepatic circulation. So this is very very important point. So with this thing, we have just completed all the basic metabolism points, and next two important things is your electron transport chain. In all the paper we can find a question from electron transport chain, and another one is our electron transport chain inhibitors and all. This is super important topic, and then we are entering into the thermogenin uncouplers and all will coming into play here. So next, coming to your this thing, electron transport chain is present now. See, electron transport chain, there are only 2 to 3 lines that you should be knowing. So that is your complex 1 to 4, complex 5. So complex 1 to 4, it is called as what? Oxidation of your NADH or your FADH2, that's all. Okay, but your complex 5, it is called as ATP synthesis or ATP synthase complex. So this is very very important. First point done. So second thing is, see, there is 1, there is 2, there is 3, there is 4, and there is 5. So in this direction only it is going to occur, so that everyone will be knowing. But it is a proton is pumped now, so it is something redox potential is there. So what is the potential difference means? So the starting potential in your complex 1 is your negative potential, that is your 0.3, and in your complex 5, the end potential will be your 0.8. So they will be asking what is the redox potential of your whole electron transport chain, then that is your plus 8 plus 3, that is your 1.1 to 1.14 volt. So this is your redox potential of your whole electron transport chain because of the flow of your electrons through these complexes, and this flow is mainly determined by your redox potential. That is the difference between the potential between your complex 1 and complex 5. That's all. So there is no proton is accumulated in your complex 2. That is also very very important point. So next, coming to your complex 1 name. Complex 1 is NADH CoA oxidoreductase, 2 is your FADH2 CoA oxidoreductase, 3 is your CoQ cytochrome C oxidase, 4 is your only cytochrome C. This is CoQ cytochrome C. This is only cytochrome C, and complex 5, as already told, it is your ATP synthase or ATPase. Okay, so this is very very important point. So in the complex 5, the structure is very very important. So we have alpha beta. So alpha beta is present in your matrix, and in your inner mitochondrial membrane we have your hollow subunit, that is your gamma. So what is the main function of this hollow subunit? Means that can only rotate like a motor, so it is called as rotator subunit or your gamma subunit. So this is very very important, and it is the thing that is finally aiding in your proton transport, and that is the by using that process only our ATP is getting generated. So this is very very important point. So this final process is there now, and also it is made up of two subunits, that is your F, O and Fi. That's all. And also this rotation of your gamma on its own axis due to transportation of proton through it, it induces a catalytic activity on your beta. So this rotation, it is going to result in catalytic activity on your beta subunit. So this process is called as what? Chemiosmotic theory. So this is another way of asking your question on your complex 5. So how many, if they, one more question also they have asked, 360 degree rotation of your gamma results in 3 ATP synthesis. So this is very very important. And your oligomycin. So oligomycin is an inhibitor of your complex 5 where FO is getting inhibited. FO component of your this thing is inhibited, and your because of this there is no rotational movement and there is no ATP formation, and there is your atracycloside. So atracycloside means that is your ATP, ADP, ADP antipode system, it is getting activated, and it also results in what? Both is inhibitor of your complex 5, and there is no ATP generation involved here. So that is very very important point. So next, coming to your basically ETC inhibitor, sure shot. Okay, sure shot. So it's complex 1 means what? That is your pyrisidin, your phenobarbital 1 is coming, and also rotenone 1 is coming. So this is very very important. Next, coming to your complex 2. Complex 2 is carboxin, by for your 2, carboxin, carbosin, or your TTFA. Don't confuse between your 1 here and your triethyl fluoroacetone. Next, coming to your complex 3. So that is your British anti-leucide or anti-mycin, or your anthromycin. Okay. And next, coming to your complex 4. So complex 4 is a cyanide, and the most important question for your complex 4 inhibitor, it is a carbon monoxide. Carbon monoxide can inhibit, but carbon dioxide has nothing to do with it. Okay, this is the question they have asked, James question. Okay, 17 or 18. Okay, and also your H2S gas is an inhibitor, and your cyanide is an inhibitor, and also your sodium acid is also an inhibitor. So this is your ETC inhibitor. So next, coming to your uncouplers. So uncouplers, very very important. So natural, chemical. So chemical is very very important. So in chemical, 2,4 dinitrophenol, 2,4 your dinitrocresol, and also your chlorocarbonyl cyanophenoxyhydrazone. Just remember CCCP. And also one more mycin. I couldn't remember the name. Some mycin will be coming. That is a potassium channel opener. So that also will result in your uncoupling. That is a chemical uncoupler. And this and all, it's natural. That is your thyroxine, free fatty acids, thermogenin. So these and all will be coming into your natural uncouplers. So ultimately what does this uncouplers do means they will uncouple your electron transport chain, as a result of which your potential of your H plus proton is reduced too much that it can't get excited again. So because of this, when H plus is not pumping into your electron transport chain, there is no ATP is generated. So this difference between the actual H plus energy level and your basal, now because of the uncoupled H plus, this thing is dissipated as heat energy. So that is the reason why thermogenesis is occurring. That protein is called as what? Thermogenin, and it is naturally present in our body. So this sums up our whole metabolism of your carbohydrates. Next is starting into enzymes. Okay, so in your enzymes, 5 concepts totally. So first you should be knowing your classification. So for classification, what is the mnemonic you should be remembering means that is your OTHLIL. So O for your oxidoreductases, transferases, hydrolases, liases first. Liases, synthase is a liase, synthetase is a ligase, and isomerases. That's all you should be knowing. So oxidoreductases, any dioxygenase, monooxygenase, and all oxidoreductase. Transferase, any group is transferred. Hydrolase, by using water you are breaking something. By without using water breaking means that is called as ligase. And next coming to isomerase, that epimerase. Mutase and all coming under this group, and synthetase, all these synthetases comes under your ligases. And one more point you should be remembering is your all carboxylases also comes under your ligase only. So next coming to your competitive and your non-competitive inhibitor. So in this thing, before you should be knowing, there can be two questions, two graphs that can be asked. So one graph is your Michaelis-Menten graph. So Michaelis-Menten graph means that will be this will be it. So like this only the graph will be it for your Michaelis-Menten plot. So whenever your Lineweaver-Burke plot is given means this will be the diagram. So we shouldn't miss it up. So whenever similar looking, so that is your competitive inhibition. Height is different means dissimilar, so that is your non-competitive inhibition. And your uncompetitive inhibition means, see, it will be similar but it will be just diminished in size. So that is your uncompetitive inhibition. So this is for your so this is very very important for your this thing. What can I say? You are able to hear me? This is called as what? This is your Michaelis-Menten graph. Lineweaver-Burke plot. This is whenever you can see an X that is crossing over means that is competitive. Whenever you are seeing no competitive this thing, but they are meeting at one point and diverging lines means that is your non-competitive, and your parallel lines means that is uncompetitive. That's all you should be knowing. And also suicidal inhibition. Suicidal inhibition means, or mechanism is another name. Suicidal another name is what? Mechanism based inhibition. So in this thing, what is it needed? So there is xanthine, there's the uric acid. So this is normal thing that is happening in our body, xanthine oxidase. Xanthine oxidase inhibitor is your allopurinol. Allopurinol following what? Mechanism based inhibition or suicide inhibition. Why? Because this allopurinol, in presence of xanthine oxidase, it will form alloxanthine. Alloxanthine will be inhibiting this reaction. So that is why this is called as mechanism based inactivation. So this is very very important. So next coming to your cofactor and your coenzyme. So this is classification. The classification is very very important. So cofactor or your coenzymes. So here holoenzymes. Holoenzymes means complete functional enzymes. So apoenzymes. Apo has a P, so which means that it needs protein. And another one is our cofactors. Cofactor does not have a P, so no protein is involved. So what is present? There can be vitamin or there can be a metallic enzyme. So this metallic enzymes is very very important point here. So how we are going to remember means molybdenum. So molybdenum means most of your oxidases will be molybdenum, and also your superoxide dismutase is there. Now superoxide dismutase needs zinc, it needs manganese, it needs copper. Okay, and also selenium. Selenium is also very very important. So selenium, Casson's disease and all this thing will be affected. So that is your glutathione peroxidase, deiodinase, or your thyroidotin reductase, or your glycine reductase. So this is very very important point. So next coming to your isoenzymes. So isoenzymes, this is the last concept for your enzyme here. So isoenzymes are nothing but your physically distinct form of same enzyme is known as isoenzyme. So there is a true isozyme. So true isozyme, first form is, you should be remembering, is malate dehydrogenase, where two forms are there. That is your cytosolic and another one is mitochondrial. Another one is our amylase. So amylase is salivary is present, pancreatic is present, which means that why it is called as true. They may sound similar, they may look similar, but they come from different gene. So this is the actual true isozyme here. Next coming to your hybrid. So hybrid is nothing but it is our medically important, that is your creatine phosphokinase and your LDH. Creatine phosphokinase 1 to 3 forms is present. LDH is present 1 to 3, 4, 5 forms is present. How we are going to remember this? Means you should remember first CPK. CPK, there can be a BB, there can be a BM, or your correctly it has to be written as MB, or it is your MM. So in this thing, just to start your number 1, 2 and 3, that's all. So for your LDH, it is your heart. Okay, and then change 1 M, 2 M and your 3 M. Accordingly, put the numbering here. So 1, 2, 3, 4, 5. That's all you should be knowing. And another one is isoform. So if they're asking about isoform means, so isoform, form is changed. So when form is changed, it means it will be coming from same gene, it will be similar, but your post-translational modification of the proteins will be different. So this is an example is your ALP, alkaline phosphatase. So in alkaline phosphatase, your sialic acid content differs in most of the thing that is present in bone, placenta, bile, WBC, GIT mucosa. Everything alkaline phosphatase is present, but your sialic acid content is different, and this is because of post-translational modification. Because of this, this comes under your isoform. So next coming to your isocitrate dehydrogenase. So isocitrate dehydrogenase or malate dehydrogenase, it is similar. So we'll be having two forms, that is your mitochondrial form and your cytosol forms. So by this thing, first half of the biochemistry is over.